So, well, as you know, there are a number of challenges in the field of CAR T cells, although some patients respond extremely well to this strategy. Up to now, a majority of cancer patients is refractory to such a therapy, especially in solid tumors. So really highlighting the presence of resistant mechanism that one needs to identify in order to overcome them and propose more powerful solution. And among the different resistant mechanisms, the suppressive tumor microenvironment, as we've heard from Suman Mitra, is really important. The other, another challenge, which is related also to CAR T cells, is the huge number of immune cell products which have been developed. I'm sorry, the slide is a little bit crowded, but here you could see at the center the different cells which have received the agreement, and all around you have this constellation of cells which are cell products which are either in preclinical development, phase 1 or phase 2. It could be autologous CAR T cells, allogeneic products, and immune cells equipped with a CAR. I'm sure we hear about other cell types which are very important, also NK cells, gamma delta, mate cells, and others. And the important question is, in which model can we test this novel T-cell product? Because we know that the current models are not the good one. So definitely we need more relevant models in order to test efficacy and also potential toxicities. So we at INSERM, Institut Cochin, we have been involved in the development of a new model. It's an organotypic model. It's based on fixed slices made from human primary tumors. So we have ongoing collaborations with clinicians at the Hôpital Cochin that provide us with fragments of fresh tumors. Could be, for example, non-small cell lung tumors. And this is a protocol. I won't go into the detail, but basically we cut the tissue with a vibratum. We make fixed slices really to maintain quasi-intact the tumor microenvironment. And on top of fresh slices, nothing is fixed. We add the T cells, could be T cells purified from the same tumors, the T cells, or it could be some T cells which have been genetically engineered to express a CAR. The T cells infiltrate the tissue is rinsed, immunostain to look at also the tumor cells, or stromal cells, stromal component, and then we use analytical technologies, including imaging microscopy. So we could look at, and this is some of the example, we could look at the distribution and migration of CAR T cells. So you have an example of CAR T cells in green, in human lung tumor slide. The tumor cells, it's very compartmentalized organ, as you could see. The tumor cells are in white, the stroma is in red. It's one image every 20 seconds for 20 minutes. And you could see that most of the CAR T cells are actually not in regions where we would like the CAR T to be in contact with tumor cells, but really excluded from the, sequestered into the stroma. We are also able to look at the activity of CAR T cells, focusing on the calcium response. We use specific calcium dye for that, and I'll just show you an example. When the CAR T cells, they get into contact with their target, you could see that they present a flash of calcium that we analyze using automated softwares. We could keep the explants for up to 2 days, allowing us to look at the killing abilities of CAR T cell, their cytotoxicity, and again, we use fluorescent dye. And in that pictures, you could see that the CAR T cells in red, they are associated in regions where you have many apoptotic cells. So we have used this technology over the years to really identify a number of obstacles that prevent T cells and CAR T cells to perform their job. We've, for example, identified a subtype of macrophages, and I think I have some movies there, a subtype of macrophages present in human tumors that really sequester T cells in the stroma. You could see there by doing video microscopy, but the macrophages in red, they form long-lived interaction with the T cells, really impeding T cells from contacting tumor cells. We also shown that the extracellular matrix, which is a hallmark of growing tumors, also dictates the way T cells migrate. As you could see there, the T cells, they migrate along the fibers which are parallel to the tumor stroma interface. We've also recently shown that the lack of adhesion molecules at the surface of the T cells really prevent the T cells and the CAR T cells to form an immune synapse. So now that we have identified a number of obstacles that I've just described, our goal is really try to design some innovative strategies actually to boost the efficacy of T cells, of CAR T cells. So we have a number of strategies, developed a number of strategies. We could increase the migration of T cells, like, for example, boosting the migration and the metabolism of T cells. We could obviously reprogram the macrophages, for example, neutralizing TGF-beta, targeting the extracellular matrix. Also, we have designed a number of different strategies, and we're also working on an epigenetic factor, and we have some, I think, nice data showing that we knock out this epigenetic factor, CX6A5, we could increase the persistence and the expansion of CAR T cells. So in terms of perspective, I think the platform that we have developed is very amenable for evaluating the effect of novel cellular immunotherapy. We're also doing, we plan to screen for therapeutic molecules able to restore T cell activities within tumors and within an intact tumor microenvironment. Use a slice assay also for testing the toxicity. And if we are interested in CAR T toxicity, please feel free to reach out to me. Of course, for conclusion and as a wrap-up, we have developed a relevant preclinical patient-derived tumor model to develop a number of innovative targeting strategies to improve CAR T cell efficacy. We have a strong collaboration with clinicians for obtaining fresh tumor biopsies, and we are a part of a number of national, but also international consortium, the French National Consortium UniTC, supported by INCAR and Cancer Grand Challenges on CAR T cells and pediatric solid tumors and T2Evolve also. And I think, yeah, so the work has been done at the Institut Cochin, the team Cancer and Immune Response, and I'd like to thank all of our collaborators, the clinicians, the academic, but also industrial partners, and you for your attention.